2 of microbiology portion. So today we are going to start learning about bacterial nutrition and growth. Bacterial growth is measured by an increase in cell number, whereas growth of most other organisms is measured by an increase in their size. All bacteria require a source of carbon to synthesize components of cytoplasm as well as a source of energy in order to grow. Bacteria can be grouped into nutritional classes based on how they satisfy these requirements. Nutritional aspects of bacterial growth: sources of carbon, energy, and hydrogen, electrons. Carbon source: autotrophs, heterotrophs. Autotrophs, CO2 sol or principles biosynthetic carbon source, but in case of heterotrophs, reduced preformed organic molecules formed from other organisms. Then energy sources: phototrophs, light as their energy source; chemotrophs, oxidation of organic or inorganic compounds; then hydrogen or electron sources; lithotrophs, reduced inorganic molecules act as electron source; organotrophs, organic molecules act as an electron source. Okay. Then major nutritional types of microorganisms based on the primary source of carbon, energy, and electrons. Microorganisms are placed in 4 major nutritional classes: photolithotrophic autotrophy. Photolithotrophy. Source of carbon: carbon dioxide; source of energy, light energy; source of electrons, inorganic hydrogen or electron donor. Representative microorganisms: algae, purple and green sulfobacteria, blue-green bacteria, etc. Then photo organotrophic heterotrophy. Organoheterotrophy. Source of carbon, organic carbon source, CO2 may also be used. Source of energy, light energy; source of electrons, organic hydrogen electron donor. Representative microorganisms: purple non-sulfobacteria, green non-sulfobacteria. Then chemolithotrophic autotrophy. Chemolithoautotrophy. Source of carbon, carbon dioxide; source of energy, chemical energy source, inorganic; source of electrons, inorganic hydrogen electron donor. Representative microorganisms: sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria. Then inorganic electron donor: NH3O2, NO2 minus, ammonia oxidizes, nitrosomonas eutropy, NO2 minus, O2, NO3 minus, nitrite oxidizes, nitrobacter vinogradsky, like that. Then chemoorganotrophic heterotrophy. Chemoorganoheterotrophy. Source of carbon, organic carbon source; source of energy, chemical energy source, organic; source of electrons, organic hydrogen electron donor. Representative microorganisms: protozoa, fungi, most non-photosynthetic bacteria. So here we mentioned important classes, major nutritional types. Photolithotrophic autotrophy, photoorganotrophic heterotrophy, chemolithotrophic autotrophy, then chemoorganotrophic heterotrophy. So that's all about that. And culture media: for any bacterium to be propagated for any purpose, it is necessary to provide the appropriate biochemical and biophysical environment. A culture media is a solid or liquid medium that contains all the nutrients and microorganisms required for growth. It is used to grow, transport, and store microorganisms depending upon the special needs of particular bacteria as well as particular investigators. A large variety and types of culture media have been developed with different purposes and uses. Culture media are employed in the isolation and maintenance of pure cultures of bacteria, and are also used for identification of bacteria according to their biochemical and physiological properties. Culture media can be classified on the basis of physical nature: liquid, semi-solid, and solid; chemical composition like synthetic and complex; and functional type: supportive, enriched, selective, and differential. So what is a synthetic medium? Defined medium. A chemically defined synthetic medium is one in which the exact chemical composition is known. A defined medium in a minimal medium if it provides only the exact nutrients, including any growth factors needed by the organism for growth. Then complex media, a complex undefined medium is one in which the exact chemical constitution of the medium is not known. Example of some common complex media are nutrient broth, tryptic soy broth, and makangi agar. These are complex media, undefined medium. Then enriched media. An enriched medium contains some components that permits the growth of specific type of species of bacteria, usually because they alone can utilize the component from their environment. However, an enrichment medium may have selective features. Blood agar is an example of enriched media because it encourages the growth of many fastidious microbes. So an enriched medium contains some component that permit the growth of specific type or species of bacteria, usually because they alone can utilize the component from their environment. However, an enrichment medium may have selective features. Blood agar is an example. Blood agar is an example of enriched media because it encourages the growth of many microbes. Then selective media. A selective medium is one which has a component added to it which will inhibit or prevent the growth of certain types of species of bacteria and to promote the growth of desired species. For example, bile salts in crystal violet favor the growth of gram-negative bacteria by inhibiting the growth of gram-positive bacteria. Bile salts and crystal violet. So, eosin, methylene agar, eosin, methylene blue agar, makangi agar, and mannitol salt agar are commonly used as selective medium. Then differential media. A culture medium is described as a differential medium if it allows the investigator to distinguish between different types of bacteria based on some observable traits in their pattern of growth on the medium. A culture medium is described as a differential medium if it allows. So selective medium, which we learned. Then differential media, a culture medium is described as differential medium if it allows the investigator to distinguish between different types of bacteria based on some observable trait in their pattern of growth on the medium. Blood agar is an example of differential media and is used to distinguish between hemolytic and non-hemolytic bacteria. Eosin, methylene blue agar, makangi agar, and mannitol salt agar are also used as differential media. Next is about bacterial growth. Growth is an ordinary increase in the quantity of cellular constituents. It depends upon the ability of the cell to form new protoplasm from nutrients available in the environment. In most bacteria, growth involves increase in cell mass and cell division. The sexual process of cell division of bacteria is known as binary fission. And bacterial growth curve under favorable conditions, a growing bacterial population doubles at regular intervals. Growth occurs in geometric progression 1, 2, 4, 8, etc. or 2 to the power of 0, 2 to the power of 1, 2 to the power of 2, 3, 2 to the power of 3, etc. 2 raised to n, where n is the number of generations. This growth is called exponential growth. In reality, exponential growth is only part of the bacterial life cycle and not representative of the normal pattern of growth of bacteria in nature. When a fresh medium is inoculated with a given number of cells and the population growth is monitored over a period of time, plotting the data will yield a typical bacterial growth curve. Four characteristic phases of growth cycle are recognized, like lag phase. Immediately after inoculation of the cells into fresh medium, the population remains temporarily unchanged, although there is no apparent cell division occurring. Lag phase, immediately after inoculation of the cells into fresh medium, the population remains temporarily unchanged. Although there is no apparent cell division occurring, the cells may be growing in volume or mass. An exponential log phase. The exponential phase of growth is a pattern of balanced growth wherein all the cells are dividing regularly by binary fission and are growing by geometric progression. The cells divide at a constant rate depending upon the composition of the growth medium and the conditions of incubation. The rate of exponential growth of a bacterial culture is expressed as generation time. Also, the doubling time of the bacterial population. Generation time is defined as the time t per generation and number of generations. Then stationary phase. Lag phase, log phase or exponential phase. Then stationary phase. Exponential growth cannot be continued forever in a batch culture. A closed system such as a test tube or flask. Population growth is limited by 1 or one of three factors like exhaustion of available nutrients, accumulation of inhibitory metabolites or end products, exhaustion of space. In this case, called the lack of biological space. Here we are using a closed system, remember that, like a test tube or flask. So biological space will be limited. Then death phase. If incubation continues after the population reaches stationary phase, death phase follows, in which the viable cell population declines. During the death phase, the number of viable cells decrease geometrically or exponentially, essentially reverse of growth during the log phase. So here we could see this typical growth curve for a bacterial population, and for CSIR net this question usually comes where a different, you know, representation of this particular bacterial population curve will be there, and some questions related to that will be present. So carefully learn this. Growth phase. Here we could see the x-axis represent time, y-axis represent log 10 viable organisms per ml, and different phases are marked: lag phase, exponential phase, then a stationary phase which shows turbidity, optical density, then death phase. Death phase is, some kind of lag phase. Optical density is also shown. Calculation of generation time. The generation time or doubling time is the time interval required for the cells or population to divide. When growing exponentially by binary fission, the increase in a bacterial population is by geometric progression. If we start with one cell, when it divides, there are 2 cells in the first generation, 4 cells in the second generation, 8 cells in the third generation, and so on. G generation time is equal to T by T, that is time in minutes or hours by N, number of generations. Exponential growth, Nt is equal to N0 into 2 raised. Then arithmetic plot is shown. Semi-logarithmic plot is shown. Exponential growth of unicellular organisms, arithmetic and semi-logarithmic plots of cell number versus time. A plot of the number of cells against the time gives an exponential curve. Such an arithmetic representation of a growth curve is however unsuitable for dealing with a large number of cell divisions. If it is preferable, therefore, to use a semi-logarithmic plot, straight line in this case. Such as a logarithmic plot shows exponential growth as a straight line, the slope of the line corresponding to the division rate. The steeper the slope, the greater the division rate. Because exponential growth is characterized by linear relationship between time and logarithm of cell number, it is also referred to as logarithmic growth. Calculation of number of generations. So, such a logarithmic plot shows exponential growth as the straight line, with the slope of the line corresponding to the division rate, and steeper the slope, the greater the division rate. Because exponential growth is characterized by a linear relationship between time and logarithm of cell number, it is also referred to as logarithmic growth. Calculation of number of generations. If n0 is equal to number of bacteria at the beginning of a time interval, n is equal to number of bacteria at the end of the time interval, and n is equal to number of generations, number of times the cell population doubles during the time interval, then n is equal to n0 into 2 raised to n. This equation is an expression of growth by binary fission. Applying logarithms, this becomes log n is equal to log n0 plus n log 2. And for the number of cell divisions, n is equal to n log n minus log n0 by log 2. N is equal to log n minus log n0 by 0.301. N is equal to 3.3 log n by n0. Continuous culture of bacteria. In batch culture, the nutrients are not renewed, so exponential growth is limited to a few generations only. Bacterial cultures can be maintained in a state of exponential growth over long periods of time using a system of continuous culture designed to relieve the conditions that stop exponential growth in batch cultures. Continuous culture in a device called a chemostat can be used to maintain a bacterial population at a constant density, a situation that is in many ways more similar to bacterial growth in natural environment. Then, continuous growth of bacteria. Studying the growth of bacterial populations in batch or continuous cultures does not permit any conclusions about the growth behavior of individual cells because the distribution of cell size and hence cell age among the members of population is completely random. Information about the growth behavior of individual bacteria can, however, be obtained by the study of synchronous cultures. Synchronized cultures must be composed of cells which are all at the same stage of the bacterial cell cycle. Measurements made on the synchronized cultures are equivalent to measurements made on individual cells. Then measurement of bacterial growth. For unicellular organisms such as bacteria, growth can be measured in terms of two different parameters: changes in cell mass and changes in cell numbers. Methods for measurement of cell mass. Methods for measurement of the cell mass involve both direct and indirect techniques. Direct physical measurement of dry weight, wet weight, or volume of cells after centrifugation. Direct chemical measurement of some chemical component of the cell such as total N, total protein, and total DNA content. Then indirect measurement of chemical activity such as rate of O2 production or consumption, CO2 production or consumption, etc. Turbidity measurement. Employ a variety of instruments to determine the amount of light scattered by a suspension of cells. Particulate objects such as bacteria scatter light in proportion to their numbers. The turbidity or optical density of a suspension of cells is directly related to cell mass or cell number after construction and calibration of a standard curve. The method is simple and non-destructive, but the sensitivity is limited to about 10 raised to 7 cells per ml for most bacteria. Methods of measurement of cell number. Measuring techniques involve direct counts visually or instrumentally and indirect viable cell counts. Direct microscopic counts are possible using special sites known as counting chambers. However, only dense suspensions can be counted, but samples can be concentrated by centrifugation or filtration to increase sensitivity. Electronic counting chambers count chambers and measure size distribution of cells. Indirect viable cell counts, also known as plate counts, involve plating out or spreading a sample of a culture on a nutrient-agar surface. A sample or cell suspension can be diluted in a non-toxic diluent, water or saline, before plating. If plated on a suitable medium, each viable unit grows and forms a colony. Each colony that can be counted is called a colony forming unit, CFU, and number of CFU is related to the viable number of bacteria in the sample. Influence on environmental factors on growth. Growth of microbes are greatly affected by the chemical and physical conditions of their environments. Many environmental factors like temperature, pH, water availability, and oxygen play major roles in controlling growth of microorganisms. The effect of microbial response to major environmental factors and representative examples are given. Halophile, acidophile, alkalophile, psychrophile grows at 0 degrees Celsius and has an optimum growth temperature of 50 degrees Celsius or lower. Halophile requires high levels of salt concentration. Acidophile growth optimum between pH 0 and 5.5. Alkalophile growth optimum between pH 8 and 11.5. Psychrophile grows well at 0 degrees Celsius and has an optimum growth temperature of 50 degrees Celsius or lower. Then psychrophile grows well at 0 degrees Celsius and has an optimum growth temperature of 50 degrees Celsius or lower. Then psychotroph, mesophile, thermophile, hyperthermophile, obligate aerobe, completely dependent on O2 for growth. Facultative anaerobe, does not require O2 for growth but grows better in its presence. Aerotolerant anaerobe, obligate anaerobe, that means it does not tolerate O2 and dies in its presence. Clostridium is like that. Microaerophile requires O2 levels below up to 10% for optimum growth. Microaerophile. Bacterial chromosome and plasmid nucleoid. The bacterial nucleoid, which contains the dsDNA fibrils, lacks a limiting membrane. The bacterial chromosome is a single continuous circular DNA molecule. Bacterial chromatin does not contain basic histone proteins, but low molecular weight polyamines and magnesium ions may fulfill a function similar to that of eukaryotic histones. A bacterial nucleoid, which contains the dsDNA fibrils, lacks a limiting membrane. The bacterial chromosome is a single continuous circular DNA molecule. Bacterial chromatin does not contain basic histone proteins, but low molecular weight polyamines and magnesium ions may fulfill a function similar to that of eukaryotic histones. And plasmids, term coined by J. Lederberg, are replicants that are maintained as discrete extra chromosomal genetic elements in bacteria, also present in some yeast and other fungi. They are usually much smaller than the bacterial chromosome, varying from less than 5 to more than several hundred kbp, through plasmids as large as 2 mbp occurs in some bacteria. Replicate independently of the chromosome. Most plasmids are supercoiled, circular, double-stranded DNA molecules. But linear plasmids have also been demonstrated in genera Borrelia and Streptomyces. Plasmids can be classified into conjugative and non-conjugative type. Conjugative plasmids code for functions that promote transfer of the plasmid from the donor bacterium to other recipient bacterium, but non-conjugative plasmids do not. Conjugative plasmids are genes for the synthesis of PD that help in the process of conjugation. The best studied conjugative plasmid is the F factor or F plasmid of E. coli. The bacterial cells contain F factor are described as F star or plus F positive. F positive cells act as donor, whereas F negative cells, a cell lacking an F negative plasmid, act as a recipient. The average number of molecules of a given plasmid per bacterial chromosome is called its copy number. The average number of molecules of a given plasmid per bacterial chromosome. The average number of molecules of a given plasmid per bacterial chromosome is called its copy number. Some plasmids are present in the cell in only 1 to 3 copies, whereas others may be present in over 100 copies. Large plasmids are often conjugative, have small copy numbers, code for all functions required for their replication, and partition themselves among daughter cells during cell division in a manner similar to bacterial chromosome. Plasmids smaller than 7.5 kilobase pairs usually are non-conjugative, have high copy numbers, rely on their bacterial host to provide some functions required for replication, and are distributed randomly between daughter cells at division. Many plasmids control medically important properties of pathogenic bacteria, including resistance to one or several antibiotics or production of toxins. Many plasmids confer antibiotic resistance onto cells that contain them and are described as R plasmid, resistance plasmid. Some R plasmids have only a single resistance genes, where others have many. So many R plasmids are conjugative plasmids. Cold plasmids are responsible for the production of bacteria such as colon, a toxic protein. Type of plasmid: resistance, antibiotic resistance, fertility, conjugation and DNA transfer between bacteria, killer, synthesis of toxins that kill other bacteria, degradative enzymes for metabolism of unused molecules, virulence, pathogenicity. Functions encoded by plasmids. Depending on their size, plasmids can encode a few hundred of different proteins. However, plasmids rarely encode gene products that are essential for growth, such as RNA polymerase, ribosomal subunits, or enzymes of the tricarboxylic acid cycle. Instead, plasmid genes usually give bacteria a selective advantage under only some conditions. Gene products encoded by plasmids include enzymes for the utilization of unusual carbon sources such as toluene, resistance to substances such as heavy metals and antibiotics, synthesis of antibiotics, and synthesis of toxins and proteins that allow the successful infection of higher organisms. bacteriosin which kills E. coli, degradation of toluene and benzoic acid, tumor initiated in plants, degradation, nodulation of roots of legume plants, resistance to ampicillin, tetracycline, and clindamycin. Antibiotic resistance, phylosynthesis, tumor formation in plants, nitrogen fixation in plants, oil degradation, gas vacuole production, insect toxin synthesis, plant hormone synthesis, antibiotic synthesis, increased virulence. Then HFR formation. The effector is an example of an episome. An episome is a plasmid that can exist autonomously in a cell or can integrate into the bacterial chromosome. A bacterial cell that contains an integrated effector is referred to as an HFR cell. The frequency of insertion occurs at about 10 raised to 5 to 10 raised to 7 per generation. Integration occurs homologous recombination between two covalently closed circular DNA molecules forming one circular molecule containing both of the original DNA structures. It is thought that the insertion sequence present in the F genome and those in the host chromosome serve as regions of homology for the insertional event. Then genetic recombination. Genetic recombination in bacteria involve transfer of genetic information from a donor to a recipient and result either in substitution of donor alleles for recipient alleles or addition of donor genetic elements to the recipient genome. Transformation, transduction, and conjugation are parasexual processes that use different mechanisms to introduce donor DNA into recipient bacteria. Because donor DNA cannot persist in the recipient bacterium unless it is a part of replicon, recombination between donor and recipient genome is often required to produce stable hybrid progeny. Then recombination is most likely to occur when the donor and recipient bacterium are from same or closely related species. Transformation. Discovery of transformation. Transformation was the first mechanism of gene exchange in bacteria to be discovered. In 1928, Fred Griffith found that one form of the pathogenic pneumococci, now called Streptococcus pneumoniae, could be mysteriously transformed into another form. Griffith's experiment was based on the fact that S. pneumonia make two different types of colonies, one pathogenic and the other non-pathogenic. Because pathogenic strains excrete a polysaccharide capsule, they make colonies that appear smooth on agar plate. Apparently, because the capsule allows them to survive in a vertebrate host, these bacteria can infect and kill mice. However, if colony-forming mutants that cannot make the capsule that are not pathogenic in mice sometimes arise from some smooth colony formers. In his experiment, Griffith mixed dead S. pneumonia cells that made smooth colonies with live non-pathogenic cells that made rough colonies and injected the mixture into mice. Mice given injections of only the rough colony forming bacteria survived, but mice that received a mixture of dead smooth colony formers and live rough colonies formers died. Furthermore, Griffith isolated live smooth colony forming bacteria from the blood of the dead mice. Live type R, monopathogenic. Live type S, pathogenic. Live type S, heat killed type S, non-pathogenic. None. Mixture of live type R and heat killed type S, pathogenic. Live type S. Including that the dead pathogenic bacteria gave off a transforming principle that changed the live non-pathogenic rough colony forming bacteria into the pathogenic smooth colony form. He speculated that his transforming principle was the polysaccharide itself. About 16 years after Griffith did his experiments with mice, Oswald Avery and his collaborators used the in vitro system to purify the transforming principle and showed that it is DNA. The work of Avery and colleagues helped show that DNA, not proteins or other factors in the cell, is the hereditary material. Process of transformation. Transformation is a mechanism of genetic transfer between bacteria in which the donor DNA is cell-free DNA in the recipient bacterium's immediate environment. DNA can be naturally released into the environment when cells die and subsequently rise. Once the free DNA is brought into cell, it may become integrated into the recipient's genome by homologous recombination. Recipient cells that undergo this process and require a new phenotype as a result is said to be transformed. This phenomenon allows only the uptake of DNA from same or related species. Transformation as discovered in Streptococcus pneumoniae and occurs in other bacterial genera including Haemophilus, Neisseria, Bacillus, and Staphylococcus. Mechanism we'll be learning with the help of next video. Thank you so much.